We'll now begin the session. Good evening, everyone, and thank you very much for joining in this session today. This is our 10th talk in the Only One Analytical Technique talk series. And the session for today is based on the role of cell-based functional assays as a critical quality attributes. This talk is hosted by the Analytical Services at Venture Center. Before we, move into the talk details, I would like to introduce and give a brief introduction about Venture Center. We are India's leading inventive enterprise incubator. We are a Section 8 not-for-profit organization, and we are hosted by CSR National Chemical Laboratory. And we were the first incubator to be approved by the CSIR. We are approved by various government entities like NSTDB and BIRAC, which are supporting agencies. So Venture Center aims to provide a complete ecosystem for deep science and tech-based startups. So what comprises about this ecosystem? So we provide mentoring services. We connect the startups to various funding agencies. Now, since these are deep tech and science-based startups, dedicated lab facilities and shared lab facilities forms the heart of this ecosystem. So we offer infrastructural facilities. Then we have various funding databases available with us, and various other partners which help in design, prototyping, trial manufacturing, so on and so forth. Coming to the scientific and analytical facilities, so we have the plug and play labs where we have like 100 plus workhorse kind of equipments, which is not only available for startups, but anybody who needs these services, be it academic labs, be it researchers or industry, all of them can use these facilities. So we operate as an open access facility. Then we have a very good prototyping facilities where the initial proof of concept devices can be made. We have equipment like 3D printers, laser cutters, laser scanners, all available with us. Then coming to the testing and analysis horizontal. So we have various core analytical equipment and certain advanced analytical testing centers, details of which I will be mentioning in the upcoming slides. So together with these services, we try to support the startups in their initial phase of their entrepreneurial journey and help take their products to market. But that said, many researchers and even big industry players have availed of our services and have benefited from it. In addition to this, we offer even the regulatory services and the tech transfer and IP related services. And as mentioned, we are supported by various national programs and schemes. So coming to the analytical services at Venture Center, as I mentioned, we have certain core analytical equipments which include HPLC, GC, FTIR, particle size analysis. Then we have certain high-end instrumentation facilities, like we have a Center for Applications of Mass Spectrometry, where we provide only mass spec-based services. And then we have like a cell studio where we do the cell-based assays. Details of the talk today is based on those assays that we provide. As I mentioned, these are open access facility. Anybody who requires these services can reach out to us, and we'll be happy to assist, all of y'all. And we provide special discounts to startups and students. All our standard services, their terms, conditions, and pricings are listed on our website. So you all may visit it for the further information. The most recent or new facility at Venture Center is the Center for Biopharma Analysis, or CBA as we call it. This is a GLP compliant facility and NABL accredited facility. So CBA is a one-stop destination for the characterization of biopharmaceuticals. Again, we are open access facility. We are GLP compliant, so we follow all the OECD GLP guidelines, and we have a dedicated quality assurance unit that helps us maintain our compliance. We are NABL accredited, so we have the NABL certification in place. In addition to the routine testing services, we also provide hand-holding and advisory support to any researcher or technology developers. And this facility has been supported by the National Biopharma Mission of the Government of India. Again, CBA has its website. All our service offerings regarding the characterization of these biomolecules are listed there. And if you need any further information, we'll be happy to provide to you all. So what exactly are the services that we provide at CBA? So we provide both structural analysis and functional analysis. Both these are provided under one roof, and very few facilities in the country do this. So on the structural side, right from the primary structure estimation to the higher order structure analysis, all the services are provided. And most recently, we are happy and proud to mention that we have developed a host of functional assays and even validated the same. And these are now available for commercial offerings. So just to mention a few: ADCC assay, CDC assay, cell proliferation, cytotoxic assay, reporter gene assays. All these are are forming a part of our routine service offerings, and our team has developed very well developed and well established validated assays, and these are being now on our offering list. And all the equipment and the software that is required for all the statistical analysis is available at Venture Center. So if anybody needs any further information, I'll be putting the details in the chat box, so you all can please have a look at the same. Just to give a glimpse of all the equipment that we have at Venture at the CBA facility at Venture Center. So we have one dedicated HRMS system. We have FTIR, then we have SPR Bioco T200. We have a capillary electrophoresis system, a high-end one, so we can carry out various assays with the same. We have fluorescence spectrometer, SEC-RI MULS, UHPLC, CD spectrometer, nano DSC, plate readers, and stability chambers. And as this is part of a GLP facility, all these equipments are IQ, OQ, PQ qualified. We have all the 21 CFR compliant software in place, and our team ensures both the technical quality and the regulatory compliance. So with that brief.
We are not able to see any of your slides.
Okay. So is it visible now?
No.
Oh, okay. So just give me a minute. I'll just share it again. So is it visible now? Are my slides visible?
Yes.
Okay. So with that, I wrap up the brief introduction about Venture Center and our service offerings, and I'll move to the session for today. As I mentioned, this is our 10th talk for our Only One Analytical Talk series. And it is an honor to have with us as speaker for today, Dr. Arumugam, who is the Managing Director and the Chief Scientific Officer at Affigenix. Dr. Arumugam needs no introduction, but just for the benefit of all our participants today, I'd like to mention he's a protein biochemist and a cell biologist with almost 24 years of experience in academic, government, and corporate research environments, not only in India, but globally. And he has a proven track record of leading and managing, developing biotherapeutics for treating and diagnosing different diseases. He has in-depth experience in preclinical development, immunoanalysis, immunogenicity, PK, CMC assays applied to drug development, with excellent understanding of large molecules, biologics, biosimilar drug development. His core strengths include bioanalytical and biomarker assay development, preclinical and clinical PKD, phage display, and developing 3D in vitro blood-brain barrier modules. In addition to this, Sir has in the last 22 years authored and co-authored several publications in leading scientific international journals, conferences, and book chapters. He is also author of more than 10 patent applications and several invention reports. He is an distinguished speaker at various international biosimilar and immunogenicity conferences, and he's also a member of the Global Bioanalysis Consortium for Large Molecules Harmonization Teams, which focus on ligand binding assays. So with that brief introduction, I'll wrap up my brief introduction to Venture Center. So for any of your queries related to any analytical testing requirements, not only for the routine analysis, but for any specialized custom offerings, you can reach out to us. Our details are listed here. I'll also be putting it in the chat box. We are also on social media, so please do follow us. I'd like to mention that this talk is being recorded and it will be uploaded on our YouTube channel. All the previous sessions have also been recorded there, so you all may view it. And all your queries can be sent on this, QR code. Please scan it and you can reach out to us. Yeah, and at the end, we'll also be taking your feedback. Do provide it to us because it helps us in enhancing this series in the coming sessions. So with that, a brief introduction. I will now hand over the floor to Dr. Arumugam. Sir, over to you.
Thank you. Can you see my slides?
Sir, not yet.
Okay. Can you see my slides?
Yes, it is now visible, sir.
Thank you, Edna, for the kind introduction, and thank you, the Venture Center, for giving me an opportunity to speak in this forum. As you all know, my topic is the role of cell-based functional assays, critical quality attributes. So let's move on to the next slide. So basically, I'm going to cover on because I was expected to talk on biosimilar therapeutic antibodies. That's basically most of the companies in India are developing either antibodies, monoclonal antibodies, or insulins. The predominant biosimilars that are being developed in India. So I'll give a brief background on biosimilar and then what are the bioassay definitions, key references, and also cover something on the timeline which you have to do it and what are the regulatory expectations by FDA as well as EMA and CDSC also, and some of the critical quality attributes related to MABs and insulins, and EMA's expectation for insulins, and what are the points to consider, what are the precautions you have to take for developing cell-based assays. I moved on to the next slide. I hope you can see this. So basically, overall,
yes, sir,
as of 2025, since 1985, around 215 novel biologics drugs have been approved by FDA as well as EMA. And since year 2000, over 100 biosimilars have been approved by the Central Indian CDSCO organization. And it could be insulins and monoclonal antibodies comprising parathyroid hormone as well. And in terms of EMA, as of 2022, 86 biosimilars, and in 2024, there are 24 new biosimilar recommendations have been made by EMA, and FDA is lagging behind in approving. So far, they approved only 72 biosimilars, including those with the interchangeability designations for Biocon. And what is the main difference between the biologics or novel biologics as well as a biosimilar? They follow two different pathways. One is the 351A, which is for the novel products or the innovator products, which is a very long and tedious procedure where you have to go through preclinical development and phase 1 to phase 4 clinical trials. Whereas the biosimilar product development is a very short, relatively very short compared to the novel products, and which follows the 351K pathway, which may not require preclinical studies, and it's a very abbreviated clinical trials is required, not extensive studies required. So before I go into, I just want to make sure the bioassay definition, so which everybody is aware of it. The bioassay is basically a scientific method which basically determines the biological activity and the potency of a substance, comparing its effect on a living systems. It could be cells, tissues, enzymes, or whole organisms in vivo with that of a standard preparation under control conditions. Which is basically in vitro as well, cover the in vivo studies as well. Typically, it's a definition for a bioassay. And there is always a confusion between a biological activity and a biopotency assay. Basically, biological activity tells you what a drug does, and in simple terms, and what a potency assay tells you how much of the drug is required to do what the drug does. So if you were to compare the nature of it, it's biological activity is a qualitative and a functional, whereas a potency assay is a quantitative measure. And the purpose of biological activities detects the substance works biologically in systems like a in vitro system, and and potency assay basically measures the how strongly it works. Measurement is very subjective in terms of activity, whereas potency assay is very standardized and all relatively in relation to a reference. And output is like a presence or absence effect, whereas it's like a in case of potency, it's a dose dependent study. Dose response curve and potency units are basically reported. Where does the bioassay comes into play? It comes into for product release, comparability studies, stability testing, process development, process characterization, process intermediates, product integrity, qualification of reagents. These are the various and ways the bioassay is being used in biosimilar development as well as in the biologics, novel biologics. So there are USP chapters related to the biological potency test determination. 111, which is basically analysis of biological assays, and 1030 basically gives you the definitions of all the bioassays, and 1032 is a design development. 34 gives you biological assays. 1033 gives you the validation of the assays. There is a specific 121 which gives specifically for insulin assays. These are basically insulin USP chapters which you can refer to for any potency assay or bioassays. Here I have given a lot of key references, which is basically required for biosimilar development, or many of the definitions you should be aware of it. And especially for insulins and monoclonal antibodies, it also covers some of the FDA guidance and EMA guidance. And I strongly recommend whoever is doing it for any quality attributes, like critical quality attributes, to refer to any drug. If you are developing adalimumab or glargine for insulin, in that matter, you can look at the European Public Assessment report of that particular drug. For example, adalimumab, there are 7 different reports are available where you can refer to different quality attributes which have done by different companies to get their approvals. Likewise, for FDA, the CEDAR product quality reviews basically summarizes the table. In addition to all the details, the quality attributes which they have done in terms of structural data, which method they have used, as well as the functional assays they have used for cell-based assays, and everything will be listed, which you can refer to. And you may have to relatively, if you want to develop the product for US FDA market or the EMA, you may have to literally have to reproduce this data in comparison to the reference product. What is mainly missing in any of these guidelines or white paper is the format of assays is not recommended, number of assays to be done, type of assays, sensitivity expected, new assay implementation, and quantification parameters. These are all missing, which you may have to refer to many papers. You may have to seek consultant advice, or you may have to go to FDA and EMA before you start the work, and make sure that you are using the right assay, right cell lines, and right readout you are using just to demonstrate the biosimilarity. So the cell-based assays in biological development, I just want to say, it is not only used in biosimilar development, it is used extensively for novel biologics, as well as like monoclonal antibodies, where the cell-based assays like ADCC, mechanism of action like ADCC, CDC, ADCP, receptor binding signaling pathways are used. Now the new living cells like cell and gene therapies, like a CAR T cell therapy and all, like you still have to demonstrate some of the assays for like a viability, proliferation, differentiation, transgene expression. And as well as in vaccines like immune cell activation cytokines. Typically, the readout will be, either you use a cells, primary cells, immortal cells, or engineered cell lines are used for receptor expression to find the drug antigen interaction or drug ligand interaction. And for antibodies, it's ADC, CDC assays, and some primary cells are used for proliferation and inhibition assays. And engineered cells have been used for signaling pathways and activation assays using some specific genes as luciferase gene, which is used for cell-based assays. When and where you develop the cell-based assays or the bioassays? Typically, if you are doing a novel biologics, it starts from drug selection, like a drug discovery onwards. where the cells are used upfront. If you were to develop a phage display technology and you want to develop an antibody, it is not only use recombinant antigen for your drugs, selecting the drug candidates. It's advisable to start selecting this drug from a screen and select the drugs right from cell-based assays, either by FACS sorting or by ELISA method. Whereas in biosimilars, since its timeline for development is very short, process starts from cell-based characterization for the drug expressing cell-based characterization, preclinical development, clinical development. So this, since it's an abbreviated pathway and you don't have to do preclinical or some of the clinical reduced, you still have to demonstrate the functional assay and cell-based assays, which is relatively extensively. You have to do it not only for your biosimilar product, you also have to do it for the innovator product. It is always a head-to-head comparison. We have to do it, and so parallelly, when you start doing the drug process development, you may have to work on developing the cell-based assays and identifying, characterizing, and validating the method before you start using it in your demonstrating the biosimilarity. I just want to show some data about the antibody and structure. So I'm sure everybody knows that antibody is considered as the magic bullet, and it is a drug which makes wonders. Like so far, around 106 monoclonal antibodies have been approved by various regulatory agencies, and typically it is a very functional and it's the second predominantly highest amount of protein present in your serum, next to buoyant serum albumin. And so there are two regions. One is of the Y, which is basically the heavy and light chain, of which the Fab region, which is basically responsible for binding to the target antigens, and FC region, which is basically provides the responsible for extending the half-life of the drug. Typically, a monoclonal antibody is half-life in serum, or the antibody serum half-life anywhere ranges from 15 to 21 days, and the FC region of the antibody is primarily responsible by binding to FcR and protein. So, so far, as I mentioned, there are various forms of various antibody classes are available. There are 5 different classes, of which 4 out of 5 classes of drugs have been approved, or one of them, 3 of the classes of drug have been approved by FDA, and one is in phase 3 clinical trial. Among all the 5 classes, IgG, one being the predominant one, so far 106 monoclonal antibodies have been approved as a drug, and the major subtypes being IgG1, and the next is IgG2 and IgG4 out of the 106 have been approved. In IgM, there's only one antibody, pentaglobulin, and for IgA, which is in phase 3 clinical trial, and IgE and the Xolair, which is actually omalizumab, which is used for asthma treatment. That's the only drug approved in IgG class. Nothing in IgD, which is not relevant and is not a very predominant class in the in vivo system. As I showed you the structure, the Fab function is mainly involved in target binding. And so the attribute being the Fab region, this complement determining region of the light and heavy chain, bind to the target, and that gives the specificity of the antibody to the specific antigen. And typically the binding characterization is done using flow cytometry by binding to cells expressing the target antigen, and as well as for by other methods like this SPR, which is basically Beacore, and ELISA and BLI and mesoscale discovery assays are used for target binding. These are some of the binding assays, in vitro assays, which is required to characterize your antibody binding to the antigen, where you determine the target affinity, demonstrate the affinity of the antibody in relation to the innovator product. Besides that, the in vitro assay as well, like potency assays like proliferation, neutralization, receptor, like whether it's an agonist or an antagonist, whether it's an inhibitor, that all demonstrated in a cell-based assay as well. For both Fab and FC functions, cell-based assays are used. The purpose of Fab function is demonstrate the binding and demonstrating the activity of the assay, whereas the FC region of the antibody basically is involved in the actually immunoprofiling. Basically, it binds the FC region of the therapeutic antibody binds to various receptors, and the binding of the receptor to recombinant Fc gamma receptors, there are various forms are available, and you have to demonstrate binding of your Fc drug developed biosimilar to binding to this one using SPR, ELISA, and various different platforms similar to what you use for Fab binding. This is just through the Fc region of the therapeutic antibody. And likewise, the cell-based assay, potency assay, this is actually a functional where you demonstrate the mechanism action of the drug. Methods like ADCC, CDC assay, and it's a CDC assay is basically nothing but the complement cascade which you have to follow. And this is almost all therapeutic antibodies follow either the ADCC or CDC assay or both. In some cases, ADCP, which is a phagocytosis assay. As I mentioned earlier, like the mechanism of naked monoclonal antibodies, the ADCC assay, which is and the CDC assay, receptor blockade, and immune checkpoint assays, which are all the new drugs. It's nothing to do with the biosimilars yet. Mostly for all the biosimilars which are approved in the market, they either follow the ADC or CDC assays, or act as an agonist or antagonist or an inhibitor assays is basically used for cell-based assays. Other drugs which have been monoclonal antibodies which have been approved and which are now being repurposed or used as a conjugated monoclonal antibodies for delivery of payloads, like to the payload act like a toxic thing for delivery into cells, target cells to be killed. That's a new things like drugs like Herceptin, which are all conjugated for treatment of breast cancers. The drug could be chemical or immunoconjugates, which is basically radioactive treatment, immunotoxins, and even siRNA or some suppression of to suppress target genes are used for delivery of siRNA. Now the new living cells like CAR T cell therapy and bispecific antibodies, where bispecific maps, there are two arms, one binds to one target, the other one binds to different cells. Bispecific, trispecific, and different specific antibodies are being developed. Again, all the antibodies, the specificity and the versatility of the maps are being exploited to develop many different drugs, and new drugs are coming into the market. So, as I mentioned, there are different classes of IgGs, and earlier we used to switch from mouse to human, and various classes like IgG to A and D, or different types also been switched. These are all using antibody engineering techniques. As of today, there are, besides the IgG, full intact IgG, 106 maps. There are various fragments around fragments, Fc fusion protein, ADC, drug conjugates, bispecific antibody mixtures, immunoconjugates. There are 158 antibody-based drugs have been approved so far, and it is already in the market. And this, many of you may be familiar with this. So the only human antibody first came into existence. The first one which got approved is the adalimumab. Humira is the first human antibody, which is again a phage-derived and antibody, the first drug which to be approved. Earlier, in 1985, the mouse monoclonal antibody was used as an antibody therapy, and because of the immunogenicity, it is withdrawn in the year 2010, I think. And then engineering of the antibody took place. Chimeric antibody, where 66% is only human, rest of them is a mouse sequence. So mouse monoclonal antibody was discovered as a drug, and later on it was converted to as a chimeric antibody by engineering techniques, antibody engineering. That's the popular drugs are Maptera and rituximab and infliximab. Because of the immunogenicity issues of the heavy and light chain framework regions, Then further refinement, improvement happened with the subsequent drugs, like where 90% of them are human antibodies, human sequences. Only the mouse CDR alone is from the mouse. Relatively, when they moved from mouse to chimeric to 90% human, the immunogenicity issues were resolved, the safety issues were resolved. Thing. Now, most of the fully human monoclonal antibodies literally doesn't have or no immunogenicity issues. What are the regulatory expectations on a functional assay? Basically, there are different guidelines are available. I don't want to go into details, the different ICH guidelines, which tells you, and as well as the FDA and EMA guidelines you can follow. Uh, Potency is actually it's a critical quality attributes. It requires for the biologically relevant assays. This is to demonstrate the mechanism of action of the drug, and it has to be done in comparison to the innovator product. The mechanism is it should be relevant and functional assay recommended, meaning that same target should be expressed in cells which is going to be targeted in the sensitive species of like either an animal or a human, uh, where the cells are expressing the same antigen. The ICH Q6B is basically the kind of a Bible you have to follow for all the functional assays required to confirm biological activity. And typically for any analytical or bioanalytical validation, you have to demonstrate all the methods which you develop for cell-based assay for specificity, sensitivity, accuracy, reproducibility, and in some cases like a parallelism in relation to the innovator product. Okay, what are the FDA biosimilar approval process? As I mentioned earlier, compared to the innovator product, is biosimilar is a very abbreviated procedures you have to follow. Uh, Between comparison to the innovator product, the biosimilar, you should follow the same mechanism of action, and that means you have to demonstrate in the same cell lines, same output should be produced, and the conditions of use must also, meaning disease conditions, also should match the same. Like off target, we cannot demonstrate for biosimilar approval. You have to use what the innovators have used for which same disease. You have to use your biosimilar development as well. In terms of, you have to match the route of administration, dosage form, and strength, and should be manufactured facility meeting the GMP standards for quality of the product to be for safety, purity, and potency. Extensive comparison to reference product is required. What do you mean by comparison here is there are two different things. One is the structural and functional characterization. Between the two, functional is more important in demonstrating the biosimilarity, and this forms the basis for approval of the biosimilar product, or to circumvent not doing a preclinical or extensive clinical trials. It is a structural as well as functional characterization of biosimilar in comparison, head-to-head comparison with the innovator product is a must. And so you should demonstrate high similarity, which is the most sensitive methods, like using is the most sensitive. All the comparative analytical studies forms the foundation for any approval of biosimilars, which includes structural assays for like identity, quality, safety, purity, and potency, physicochemical assays, and functional assays like a pharmacological, immunological, and additional clinical data may be required. So this is basically forms the basis for getting a biosimilar approval. In addition to that, definitely clinical trial has to be done, where you show the exposure. Basically, exposure is nothing but the pharmacokinetic assay, and efficacy is the pharmacodynamic, and safety nothing but the immunogenicity. You have to demonstrate it is as similar to the innovator product. So your design of your study for clinical as well as for cell-based assay should demonstrate that it is not deviating too much from the innovator product. Some additional clinical pharmacology studies may be required. Suppose it's for like any extra drug interaction has to be verified, that would be required if for certain diseases. And FDA or EMA may warrant additional clinical studies to approve your biosimilars, depending upon what innovators have recently done. Which is basically innovator companies are coming up with the new assays for a batch release test. So if they include a new cell-based assays, then if you're developing a biosimilar right now, you have to look at the latest certificate of analysis of the innovators, and we have to figure out what assays they are using for releasing the batch releases, and you may have to develop that one. And this is one of the experience I have seen, like because one of the drugs, the innovators, when the patent is about to expire, they did a pediatric study, and all the pediatric studies, the drug which they released, they used a new set of assays, cell-based assays, engineered cell lines were used to demonstrate, and the biosimilar companies have to develop the same assay and show demonstrate to prove the biosimilarity. Why cell-based assay matter for biosimilar? Basically, it shows the functional equivalence compared to the innovator product, and bioactivity measurement. Basically, it measures the potency or the pharmacodynamic effect in cellular responses like inhibition, proliferation, induction of apoptosis. And it is a regulatory requirement. You have no escape from that. You have to do it. And it is also ensures the quality of your control of your product. And it is applied for batch release and as well as for post-marketing surveillance to ensure it is lot to lot bioactivity and consistency. And it is to some extent the bioassays or cell-based assays are very sensitive to structural changes. It can detect subtle variation, especially for glycosylation or a protein folding, if it were missed by the physicochemical tests. Okay, now the actual topic, the critical quality attributes. Uh, what does that mean? Like it's they basically comprises of the physical, chemical, biological, and microbiological properties of the drug that directly influence the safety, efficacy, and overall quality of biologics, including attributes such as potency, purity, identity, stability, and immunogenicity. This is as per definition. And what are the physical, chemical, and biological, and microbiological? It is listed over here. And how does it work? And basically, the while you are monitoring for the clinical quality attribute, it basically guides in the design and control of your manufacturing process, especially our upstream and downstream process, and also used to assess the risk and establish the control strategies. And it play a crucial role in regulatory submission and as well as approval. Since it's the must, you have to do it. Both the structural as well as the functional assays have to be done uh, to get your biosimilarity approval, because that forms the foundation for getting approval from, especially from EMA and FDA. This is just a cartoon depicting what is an ADCC and CDC assay. So there are commercial cell lines are available for this engineered cell lines from companies like DiscoverX and Promega. And my advice is, like, if the innovators have used, or if they have used the same cell line and you are developing an assay, make sure you have a if the innovators have not used and you're going to use that one, please seek advice from the regulators before you start developing the method or using these methods. Although the readouts and everything demonstrate that since you're going to compare with the innovator product and biosimilar product, it the data may be acceptable, but it is better to get it verified or checked before proceeding with the assays if you are using commercial cell lines. And even if you're developing your own cell line, extensive characterization has to be done, and all the precautions have to be taken before because this is the mechanism of action where you're demonstrating between a biosimilar and an innovator product. So ADCC being the antibody dependent cellular cytotoxicity assay, and CDC is a complement mediated cytotoxicity assay. So I don't want to go into the mechanism here. So there are different readouts are available. As I mentioned earlier, like if the innovators have used radioactive method, you may have to use it. If you're not going to use, and if you're going to use a fluorescence or a luminous based assays, it is better to get opinion from the regulators or FDA consultants to make sure that whatever data you're going to submit and generate and submit will be accepted by regulatory agencies. The other one is like Alamar blue staining, just to show whether the whether it's live cells versus the dead cells, you can monitor, and it could be monitored by flow cytometry as well. And the other one is the enzyme assays like LDH and GAPDH, which is basically using enzyme substrate reaction and the color formation. It could be a colorimetric or a luminous or a fluorescent based assays. Dead cell staining, propedium iodide, or fluorescence staining, which is also used earlier, but now many new drugs are developing new cell-based engineering just to and file a patent to prevent others competing for those products, especially develop it just to prevent the entry or delay the entry of biosimilars into the market. So, since the cell-based assay forms the basis for approval to demonstrate the mechanism of action, it is very critical that whatever cell lines assays you do, it is similar to what is being. It is better to copy or better to find out what EPAR. You can refer to the European ones. If adalimumab, there are 7 different maps have been developed, and if you can check which one is being used by which company, and then basically you have to follow the same thing and develop and do the validation and do sample analysis and do batch release. So what are the functional critical attributes? So what I have done is there's a publication came in Pharmaceuticals in 2021. This is for adalimumab, where they have done comparison from 7 different adalimumab which have been approved. In total, there are total 77 quality attributes have done among the 7 different adalimumab approvals they obtained. In here, for the functional quality attributes, which is mainly on the left, what you see is the biological activity, or mostly cell-based assays, where the adalimumab binding to TNF alpha. There are total 24 quality attributes, out of which 13, one in bold, are potential critical quality attributes, which has to be demonstrated between biosimilar and innovator products. The immunochemical activity, where the FC region of the antibody binding to various FC receptors by in a binding assay as well as in cell-based assay. These are various functional quality attributes. So 24 here. And in terms of structural quality attributes, out of 53 structural quality attributes, which needs to be tested for your biosimilar development, which includes the physicochemical properties, primary structures, higher order structures, the most prevalent being the post-translational modification. There will be variation from batch to batch, not only in your product biosimilar development, as well as innovator product. But there are certain glycosylations which is a no-go. So we have to make sure that glycation doesn't happen, which could affect the functionality of the assay. It could have an interference in the functional assay. But it is understood that all the regulators understand it is not possible to duplicate exact glycation, because even innovators vary from in terms of post-translational modification. The one highlighted in bold are very critical, are potential critical quality attributes, which every company have to pay attention when you're developing therapeutic antibodies. Besides the enzymatic, there is non-enzymatic glycation, post-translational modification, like oxidation, deamidation, and glycation part of it has to be monitored as well. And then in terms of purity and impurities, like fragments, product-related impurities, process-related impurities also have to be monitored, which is like a HCP, HCDNA, which we didn't mention it here, but those are by default, you have to do it. And change charge variants like acidic and basic forms. So in total, like around 77, and it's not necessary that you have to do all 77, but pay attention to all the potential critical quality attributes. If innovators have done it, you have to do it. And preferably you have to follow the same method or format of the assay which is done by innovators. Innovators could have developed 18 years ago or before the, we are developing after patent expires, but regulators expect you to use the latest and sensitive method. You have to be aware of all the orthogonal methods for currently available. And so the most sensitive method have to be used. Even though it's expensive, you may have to do it. And that's there's no excuse whether innovators have done it or not. You have to do it. So that's the one for monoclonal antibodies. I took example of adalimumab. Then this is an FDA critical quality attributes from a presentation I took for insulins. It is not as extensive as for monoclonal antibodies, where glycation or the post-translational modification is very critical. It happens in monoclonal antibodies. It's expressed in mammalian cells, whereas insulins are mostly expressed in PKR as well as in bacteria, where not much of no post-translational modification happens. So in terms of complexity between insulins and monoclonal antibodies, insulin is relatively a easy product, but still you have to demonstrate biosimilarity in terms of structure as well as the functional assays. So structure not much extensive required as for maps. In bioactivity, there are mitogenic assays and metabolic assays. Um, Besides binding kinetics, you have to demonstrate in beer core and SPR, by beer core and ELISA or MSD, you have to demonstrate binding to insulin receptors expressed on the cells, and readouts have to be done. For metabolic activity, it's like adipogenesis, lipolysis assays, and glucose uptake assays is a must. Um, and insulin being treated for a chronic indication for a long period of time, the purity and is very, very critical, uh, because it's going to be dosed on a daily basis for a lifelong. So the regulators are very particular about product related or process related impurities. If it exceeds the limit, then you may not get approval. Again, you have to demonstrate biosimilarity head to head along with the innovator product. EMA's expectation of non-clinical pharmacology studies. I think there is actually a separate guideline. I have given the reference. If you are developing a glargine, I strongly recommend that you read this guideline and develop accordingly, where they have explicitly said what assays have to be done. One being the insulin receptor assay, where either you have to engineer the cell line, like insulin receptor A, insulin receptor B separately, where homo and heterodimer happens. So homodimer assay and heterodimer assay, and naturally occurring HEK29 cells, where insulin receptor A and B are automatically expressed, but you have to enrich or select cell lines and demonstrate phosphorylation of the receptors using insulins. Then metabolic assays like a glucose uptake, glycogen, lipogenesis assay, all have to demonstrate. The mitogenic activity mediated by IGF-1 receptor stimulation is not preferred. That's what is mentioned in this. That's not very critical. Out of the 5 or 6 different assays, at least 3 different assays should be performed. And again, I recommend if you're developing for EMA or FDA, you get the recommendation which what assays you're going to develop, get a pre-meeting done, and execute the experiments accordingly. This is for one of the biosimilars that got approved. What are the different assays have been done, which I had to get out of the EMA assessment report. Insulin receptor binding by Biacore, or cell lines expressing insulin receptor A, B, and then glucose uptake, lipolysis, glucose production, and mitogenic potential using different cell lines. And the methodology or the readouts, which is they have used. And basically, if you look at it, the mesoscale discovery, which is the electrical chemiluminescence, the original innovators have not developed, but actually this is if the recent biosimilar companies who got approval are using it, then FDA will expect you to, FDA or EMA will expect you to use a similar method or similar technology to demonstrate the biosimilarity with the innovator product, because these are all very sensitive methods. Just I'm going to a few more slides just to show what are the challenges and considerations In terms of bioassays, cell-based assays, it is very complex, and standardization is very difficult to do it, and you have to maintain the assay lifecycle like development, qualification, validation, and ongoing monitoring, because throughout your batch release, if you're going to use this assay, post approval also the cell-based assay is required. So you have to spend an enormous amount of time and effort uh, as you develop for your cell line which expresses your drug. You may have to do extensive characterization of the cell lines which is used for seeking approval as well for later on for a batch release as well. uh, it's a long timeline and also higher cost. uh, in vivo is costly and it's a low throughput, but cell-based assays are relatively easy. uh, and so cell lines, preferably I recommend you to go for a cell lines, either naturally occurring cell lines like tumor or immortalized cell lines, or use engineered cell lines for assay development. Primary cell line is not advisable, but in certain cases like PTH, primary cells are expected to be used. So in that case, you have to buy, and every time you have to characterize before using it in a assay. uh, Maintaining a cell bank as you do for your drug, expressing cells, you have to do it. There could be variability due to cell thawing, cell passage, medium use, cell maintenance method, and manipulation, and the analyst error as could also influence the assays. Besides the cell-based assay, in comparison, you may have to do non-biological methods like ligand binding assay. For example, Fc gamma receptor binding in Biocore or ELISA, you may have to demonstrate as well for your batch release. In parallel, again, look at the orthogonal methods. Uh, so just to show you an example, like besides developing cell lines for demonstrating your potency, the readout happens using a different, the output of the readout, the effect of the insulin binding to receptor. And if it phosphorylates, you have to monitor the phosphorylate protein. If it is a AKT cell line, AKT, or like phosphothyrolylated peptide or something gets phosphorylated, you have to measure it. That you have to use platforms like Luminex or a very sensitive method like ELISA based techniques can be used. And again, whatever method or whatever cell lines you use, there are two different. One is a cell based assay where you incubate your cells with the cells, and the readout happens in a different assay format. So both have to be validated together, and which is again, you can use engineered cell line, primary, or immortal cell lines. And binding assay is the Biocore. Like there are various substrate plasmon techniques are available. Different platforms have come, but the principle being the same. We, uh, there are new techniques. Again, one is uh, a refractometer. I think new technique is also available for binding assays besides ELISA, MSD, SPR. There's one other technique is available, but I'm not very familiar about that. I just want to point out this, like, uh, you have to be you have to be very wary about the protein concentration, because I have seen some suffer because the innovator product, they go by the label claim and they use it, and for a biosimilar they do a protein estimation. I strongly advise, I think most of you must be already aware, that any method you determine the protein concentration of your drug or any of your cell-based assay or anything, make sure the protein which you are estimating, the method is validated. You can't take the off-shelf like a Bradford or Lowry kit or a UV280 method and determine the protein concentration. For a product like hormones, like insulins and parathyroid hormones, the amount of drug you're going to use is very little. And if there is some deviation of 10 to 20% in the drug concentration determination, you could compromise the entire biosimilarity exercise. So I strongly recommend you validate two methods. One is the UV280 and other protein estimation method, and make sure you have the you are using the right extension coefficient of the drug for innovator as well as for biosimilar product. One of the reason I'm saying is because Typically, drugs for a especially innovative product, it could be a different age. Say if it's a 24 months expiry drug, like depending upon you may have your drug fresh. You're comparing with the 1 month old drug manufactured, whereas you're comparing with a 20-month old innovative product. There could be a degradation of the innovative product. So it is better you do a protein estimation and make sure the concentration which you're using is correct for in vivo study as well as for in vitro study. It's a small amount could make a big difference in because cell-based assays, you are not going to use a very huge amount of antibodies or a drug, and you're going to use little, and you have to make sure it is again the concentration is bias like similar, proving biosimilarity kind of thing. And I don't want to go into it like this. I took it out of DiscoverX, and for as you develop for your cell expressing drug expressing cell lines, you have to make a cell bank for your cell-based assay as well. Master cell bank, working cell bank, and bioassay cell, and you make sure you use the determine the passage you have to use your cells for the bioassays. If it is 4 or 5 passage, between 4 or 5 passage, don't exceed it and make an SOP, and make sure that prior to using it, what characterization have to be done, and even don't try to switch the medium, keep the media and everything constant. So that could make a lot of difference. Um, so this is just what I want to mention on the cell bank of your which you have to maintain for your cell-based assays. And what are the different points to consider in cell-based potency assays? Cell types, as I mentioned earlier, cell types, whether it's a recombinant, whether it's an engineered cell line or a naturally occurring cell line or a primary cell lines, make sure it is very stable. And traceability is important because when they come for an audit, you have to show complete record from where you procured, how did you characterize, and as you all know, the uh, what you're going to face, the problem is like mycoplasma contamination. That's very critical when you're banking it as well, like a master cell bank, working cell bank, and every time you make sure that you do a mycoplasma testing before you execute the experiment. Uh, so this is the future directions, the way the new monoclonal antibodies are getting approved. If you look at it, they are using stem cells and automated cell-based assays, and they're using artificial intelligence, uh, quality by design, and automation, semi-automation, 3D cell model, organoids are being used. If you ever want to going to copy the same innovative product, besides developing a drug, you may also adapt or develop methods, platforms similar to what is being developed. And most of the time, many of them would have been patented, like patented. Even the drug would get approved, drug patent expires, but the formulation patent or the assays which they use to get approval, if they use new technologies and uh, things, they may patent that as well, and it may not be easy to copy and reproduce the data to demonstrate biosimilarity unless regulators are very flexible in using an alternate method and orthogonal method. Uh, so just to summarize, I just gave a highlight of all the quality attributes we have to follow for antibodies as well as for biosimilar antibodies and for a product like insulins, and some of the precautions you have to take in developing cell-based assays. Um, you know, as I advise, my advice to you is, if you ever want to go to EMA or FDA, use a proper consultants, those who are experts in cell-based assays, uh, or follow USP guidelines. You may have to read extensively about the latest EPAR or the latest biosimilars that is being approved by EMA as well as FDA. You may not got all the informations, and if you have access or if you can get access to the uh, the certificate of analysis of the drug, which basically gives you sheds you light of what the innovators have done or doing it or trying to prevent you coming into the market. So you may have to be up to date to develop those kind of assays. Although the mechanism of action for antibodies are not going to change much, functional assays in terms of cell-based assays like ADC and CDC and other inhibition or agonist or being an antagonist. Uh, the technology or platform that is going to be used by innovators in the future to come to prevent biosimilars coming, there's going to be a barrier which you have to be aware of it and develop products. And thank you for listening. And if you have any questions, I'm willing to answer.
Sure. So thank you very much for taking us through that, elaborate presentation. I think in a very limited time, you were able to cover many aspects. And thank you very much for, diving into all these details. Before we wrap up, there are some questions in the chat box. In fact, there are many questions in the chat box. So we'll try to run through most of them in the time that is available. So there is a question from Disha. She says, what is used as a reference standard while developing a novel molecule, and how the potency range is assigned in that case?
See, if you're developing a novel biologics, that's a different story because as you make batches, you have to make a batch during your preclinical studies, and you characterize and you keep it as a reference. As you go for a GMP production, you keep updating and characterize accordingly. Because when you select your drug candidate, right, for a new product, like at least my experiences, when we develop a drug candidate, that's where we determine, for example, therapeutic monoclonal antibodies, if it is a mechanism of action is known, or so that becomes a basis and from which we develop. And uh, all the functional characterization gets done at the later point, but all the physicochemical characterization, the structural characterization, which is very common for all monoclonal antibodies, what glycation have to happen, which area it has to happen, and all this are very common. So you establish as you go along. That's why the drug development for a novel product, after you select a candidate, before going into all the preclinical, clinical efficacy studies and everything, it's like work in progress kind of stuff. And only when you have a GMP batch, that's where up to phase 3, the regulators are relaxed. And once you go for a phase 3 approval, that's a time you have to make sure all your reference product is set, and you make vials for comparison.
So I think she's also asking, how is potency range assigned, in this case, in case of novel molecules?
Yeah, see, I'm not an expert to comment on that one, but what happens is depending upon whether if you're developing treating a cancer, what's going to be an endpoint, and for example, if you are doing a Biocore assay, if the current treatment have an affinity in the single-digit nanomolar, you may have to go for a drug which is in the picomolar range for the same target or a different target, which can elicit the same potency in vivo. So whether the high affinity of the antibodies may be critical or not critical, we don't know. But you are trying to develop a product which is already exists for the same treatment. You have to prove it is much superior to what is already exists in the market.
Sure. So I hope, Disha, that answers your queries. Moving on to next, so there is query from Weber. So what he says is he wants to know your opinion on reproducibility and feasibility to develop compliant methods. So which technical lab should focus on SPR, BLI, ELISA, or MSD if they can spend money on only one? So
it's very difficult. Like, it's basically as far as I know, the most of the consultant, at least I talk to, they will say, hey, if the innovators have used MST, you use MST. That's what they will recommend. Because you can't say we don't have access to MST, so I did ELISA. That's not acceptable. And it's like, for them, it's you'll be safe if you use the same method what innovators have used.
Okay, sure. Yeah, so I hope, Weber, that answers your question. Moving on, we have a query from Anjali. So she says there is often confusion regarding the acceptable limits for percent recovery and percent relative potency. So what are the generally accepted ranges for these parameters during method validation or qualification of a robust cell-based assay or ligand-binding ELISA? So I think she wants to understand the difference between percent recovery and percent relative potency, and then what are the acceptable range in developing these validation criteria.
See, like, percent recovery is like, say, it's plus or minus 20% is acceptable in cell-based assays, 50% is acceptable. You can't say for your product, the recovery, or for your assay, you are 51% recovery, and for an innovator, it's like 149. That's a big difference. It's not acceptable. I think you have to be plus or minus 10 in terms of for a biosimilarity comparison, 10 to 20%. You have to have a proper justification why such deviation, and how, if your recovery is basically if you're adding a drug and you're trying to recover in terms of thing, right, what you're recovering, the recovery is within the limit, but if it's a plus or minus 20%, say one is 81 and the other is 119, of the innovators 119 recovery, whereas yours is 81, I don't think you can justify it. So you have to optimize the method so that your analytical recovery, whatever you are getting, should be within a very narrow range. So you get thing. It's on a case by case basis. So you have to say why the deviation happening. That means it is either they're going to say is you're not biosimilar.
Sure. Anjali, I hope that is clear. Maybe if you have a follow-up question, we can discuss that, in the next question that you have. I mean, I'm coming to that. So moving on, there is again a query from Disha. So she says in one of her potency assays, she observed a change in oxidation at FC site, and that is not reflecting in the potency of the receptor binding region at the fab. So is it relevant to use this assay, and what can be an alternative? So I'm not very clear of this question.
Is it happening in your innovator product also? Like, the oxidation happens with your whatever you're adding in the thing? Or
Hello, sir. This is Disha.
Yeah, Disha, I was going to tell you to unmute, and yeah, Disha, please, can you
So
please respond to what?
it is not happening with innovator, but as the stability time point increases in real time and accelerated both, the oxidation is increasing. So which leads to decreased in purity by CX method. But that change in oxidation or percent purity is not reflecting in my potency based assay. I am getting the constant potency between 70 to 130 for all stability time points. So because my oxidation is at FC region and my binding region is not at FC, so it is not reflecting in potency as well. So
your potency is, so like any ADCC, CDC is not affected?
No, it is actually not ADCC based mechanism of action. It is receptor ligand binding assay, which I am measuring through reporter gene assay.
Oh, it's like a inhibitor or agonist or antagonist kind of thing.
Yes,
right?
yes.
See, I think it's like, as long as it doesn't affect your potency, I don't think, because there is for post-translational modification glycated forms, this acceptable variations are acceptable, as long as you are not losing the stability of the product over a long period of time, because you have to demonstrate shelf life for a period of 24 months. The oxidation doesn't compromise. That means you may have to do it at a different ages to demonstrate that's not going to compromise the binding.
Yes. So it's okay if the critical quality attributes or of physicochemical data and biological data sometimes not go hand in hand, right?
Yeah, that's true. That's what, like, as I said, like the FDA or EMA understands that post-translational molecule we can't exactly duplicate. It varies from batch to batch. But if it doesn't happen in innovator product and happens in yours, you may visit your process, like, what is in the thing causes the oxidation.
Or maybe formulation.
Yeah. Yes. You may have to revisit that.
Thank you.
Sure. Yeah. I think in the interest of time, we move to the last question, which is from Anjali. Anjali, I think this is quite, if you're available, I would request you to kindly ask the question directly to sir, but I think she has some queries regarding the determination for batch release. So Anjali, are you on the call? Anjali?
Yes, I am here.
Yeah. So Anjali, please go ahead and you can ask this question regarding the, which you have put on the chat box. It will be better if you ask it directly to sir.
Yes, hello, sir. Actually, I just wanted to have like clarification regarding this. I encountered this incident a few months ago. So, if we are performing three determinations for a batch release sample or a stability sample, and if you observe that there's this one determination which is not falling into the criteria of, let's say, 80 to 120% of the recoveries, and the rest other two samples are passing, like other two determinations are passing, and then like what should be the course of action for this? So like the first one is either we can do is we can perform three additional determinations and calculate the geometric mean from all the six determination, or we should just say that particular determination was an outlier and we can just go ahead but by performing only one determination and report the like the geo mean from the three determination. So, what should be the course of action?
See, like typically it's up to your SOPs, what you have for your regulators or quality assurance team have it. Because typically regulators won't accept if you to get your statistics, you keep doing more experiments so that one failure will become minimal. I think that's not acceptable. I think you may have to revisit and find out what, and you may have to write a deviation and then repeat the experiments to make sure if you have to demonstrate three, you have to repeat three more times.
Okay, thank you so much.
Sure. Thank you. Yeah. So, yeah, there are a couple of questions, sir. If you have a couple of minutes, we can take that, or what I can do is I can collate them and I'll write it to you and then you can answer them. Let me know what works for you.
Yeah, I'm okay. I can answer them.
Sure. Sure. Maybe there are last two questions. So the next one is from Archana. She says, if PK profile for a biosimilar molecule is different from the innovator molecule, how do we determine the potency?
PK from human PK or animal PK?
Yeah, Archana, if you can please clarify on this. She says animal PK.
So see, you have to look at why it's different, because different in terms of whether there's an antibody related compromise happened with your drug. If it is not there in innovator, and because if you in PK comparability is not produced, means it is basically it's exposure of the drug in vivo and how it gets cleared. And if there is an immunogenicity, and if you are not monitoring the immunogenicity, regulators may expect you to demonstrate was there an antibody response for your product, but not for the innovator.
I think, Archana, I hope that clears your initial queries. I'm sure there will be some other in-depth follow-up question, but we can always connect separately. And last question for this session is by Shrishti. She says, what possible factors other than cell density and stage of differentiation could lead to well-to-well variation in metabolic assays for insulin?
It's actually a tough question. So, it's basically how did you plate it and other things are a thing. And, if in terms of your plating, if it's, was it in the center or it's in the edge effect is there, those things you have to monitor. Because all the cells were resuspended in the same manner and put in the wells. And if there's a difference, I strongly recommend, repeating it again, just to see that it has nothing to do with because typically all the plates, there won't be much of a difference. It is how you plate them, cells, and sometimes cells won't behave the same way. One of the things which I know for thing is, for example, if you're using NIH 3T cells for insulins, and if it is not from a single cell, clone, and you may see this difference. So ideally, you have to look for, you know, isolate single cell clones, expand it, and then use them. Don't just buy from off the shelf like ATCC and start using it. You may have to characterize for the receptor expression, make it as a single cell clone, characterize extensively, and then make a biobank and then start using it.
So, yeah, I think with that, we'll wrap up the session. So I take this opportunity to thank you, sir, so much for, in spite of your busy schedule, you have been traveling extensively, but still you obliged and accepted our invitation. And thank you for sharing your valuable inputs, regarding these assays. These are very complex assays. And in the last 1 hour, you've tried to, give us a good, glimpse of all that goes into having successful bioassay. I'm sure our participants have benefited from it. And thanks to all our participants also for attending the session. And please do provide us your feedback so that we can improvise on the further sessions. I have shared the feedback link. Please do give us your feedback. And as I mentioned, the session has been recorded and it will be uploaded on a YouTube channel, and I will be sharing the link with all of you all. So with that, we'll now wrap up the session. Thank you, sir, once again, and looking forward to working with you, on some of the future talks as well. Thank you so much, sir.
Thank you. Thank you. Thanks.
Yeah.
Thanks, everyone. Have a good
Thank
evening.
you.
Thanks. Yeah, you too, sir. Yeah.
Hello, Dr. Arumugam, Sujaya here.
Hi, Suja.
Thank you so much for this valuable information, important tips, and more importantly, generously sharing your insights with all of us. Thanks a lot, sir.
Thank you. Thanks. You're welcome.
So there are a lot of everybody is appreciating the session and saying that it was a great session. So you can see in the chat box, it was a good time attending the session. It was a great session. Loved the session. I will also be compiling the feedback and sharing with you. So I hope all of this, the participants will now be able to use in their respective research and development.